Inside you, right now, trillions of microscopic lives are at work. They feed you, defend you, and even speak to your brain. Over the next half hour, we will meet the most important of them, one by one. Who they are, what they look like, what they do, and why your health may depend on them. This is Meet Your Microbes from Neurosynapse Studio. Let us go inside. Within hours of birth, one bacterium starts moving in. In many breastfed babies, it can become a dominant early resident. Meet Bifidobacterium, the founder, a tiny rod that often branches into a Y. It does not just take up space. It eats, ferments, and reshapes the gut before almost anything else arrives. As it feeds, it turns the gut more acidic, helping hold unfriendly microbes back. Higher early levels are linked to lower allergy and eczema risk in some studies. Linked, not proof. So why does milk seem built to feed one microbe? Your mother's milk contains sugars your baby cannot digest. So why are they there? These are HMOs, human milk oligosaccharides. They pass through the infant gut, almost untouched, and arrive in the colon as food, not for the baby directly, but for the founder. One species, B. infantis, carries special genes built to consume many of these sugars. As it feeds, it gains ground in the newborn gut. Milk appears to support this partnership between baby and microbe. But in many modern babies, the founder is becoming harder to find. The most influential microbe of early life is going missing in some modern babies, and many parents never know. In some Western cohorts, B. infantis was undetectable in most infants studied. C-section birth, formula feeding, and antibiotics are associated with lower levels. Association, not blame. When the founder's seat sits empty, other microbes can move in, some carrying antibiotic resistance genes, and the gut may stay less acidic than expected. Across a lifetime, Bifidobacterium usually falls from infant dominance toward much lower adult levels. So can the founder be restored? Unlike many gut microbes, this one can sometimes be put back. Bifidobacterium is relatively easy to grow and appears in some probiotics. In one small infant trial, B. infantis persisted better when paired with breast milk or HMO support. Strain, product quality, and context matter, so this is not medical advice. The founder also pays it forward. As it ferments, it makes lactate and acetate, lowering pH and leaving those acids behind. Those leftovers become food for the next resident, the one that helps keep your gut wall calm. Faecalibacterium. The founder leaves lactate and acetate behind. The next resident lives on that chemistry and helps keep your gut wall calm. Meet Faecalibacterium prausnitzii, a long, slender rod and one of the most abundant bacteria in many healthy adult colons. It has a fatal weakness: oxygen. Expose it to open air and many cells die quickly. It lives deep in the oxygen-free core of your gut. That fragility is why this microbe is not simple to bottle. To meet it, you have to go where there is no air. So what is it doing there? It is eating. This fragile microbe survives on someone else's scraps. It feeds on lactate and acetate from earlier residents and on fiber your own body cannot break down. Scientists call this cross-feeding. One microbe's waste becomes another's feast. In lab systems, when Faecalibacterium grows alongside Bifidobacterium, it can grow better and make more of its useful products. That makes it a partner, not a freeloader. It takes leftovers and fiber, then turns them into something your gut wall depends on. What does it make? Out of leftovers and fiber, this microbe makes one of the most important fuels in your colon, butyrate. Faecalibacterium is one of the major butyrate producers in the gut. Butyrate is special because the cells lining your colon burn it as a main fuel source. Estimates suggest those cells get much of their energy from butyrate, not directly from your bloodstream. So this microbe is feeding the wall of your gut from the inside. But feeding the wall is only half the story. Butyrate also helps keep the peace. Why call it the Peacekeeper? Because butyrate does more than feed your gut wall. It helps wall cells seal tightly together and can signal the immune system toward calmer, anti-inflammatory patterns. Less leaking, less needless alarm. People with inflammatory bowel disease often have lower levels of this microbe. That is an association, Not proof it causes or prevents disease, but it is one of the most consistent findings in the field. Feed the Peacekeeper fiber, and it feeds and calms the wall back. That calm wall is wrapped in mucus. Next, meet the Gatekeeper. The calm wall you just met is wrapped in a slick mucus blanket. Buried inside that blanket is one plump oval microbe, Akkermansia. It does not simply pass through, it settles into the mucus and grazes on the lining that coats your gut wall. In many adults, this microbe makes up a small but meaningful part of the gut community, tucked close to the wall where few others can reach. A microbe that eats your gut lining sounds like a problem. So why is it known as a gatekeeper? Back to the oval grazer, nose deep in the mucus blanket. Here is the strange part, shown mainly in mouse studies. As Akkermansia grazes, it can signal the cells below to renew mucus faster than it eats. The blanket grows back thicker. That keeps goblet cells working and keeps the lining active. A busy, renewing lining can become a thicker, better sealed lining, so the wall may end up stronger than before the grazing began. But the real story is not only the eating, it is what the grazing leaves behind. Every time Akkermansia grazes, it breaks mucus down into useful chemistry. It releases short-chain fatty acids, the same family of fuels the Peacekeeper made. Those signals can soak into the wall cells below. In mice, they help feed the lining and tighten the seals between cells, the junctions that keep the gut barrier closed, fuel the wall, seal the gaps. This Gatekeeper pays rent in a currency the wall can use. So, what does a better sealed wall mean for the rest of your body? Picture the finished work of the oval Gatekeeper, a thicker, well-sealed mucus barrier with fewer gaps for trouble to slip through. A sealed wall may leak less of what can trigger inflammation. Here is the honest link. People with more Akkermansia are often associated with leaner body weight and better insulin sensitivity. Association, not a cure, and not a guarantee. One small early human trial looked promising for some metabolic markers, but it is not settled medicine. Akkermansia helps build the wall. A strong wall still needs a bouncer at the door. Next, Lactobacillus. Your gut wall is sealed tight, but a sealed door still needs a bouncer, and this one fights with acid. Meet Lactobacillus. These are straight or slightly curved rods, often linked into short chains. Here is the twist. Lactobacillus is famous, but in the colon it is usually a guest, not a major tenant. It often passes through. Its real strongholds include fermented foods and other acidic body niches. In the gut, it still works the door. So how does a passing guest defend a place it barely lives? With chemistry. A guest cannot win a turf war by sitting still. Lactobacillus eats, ferments, and weaponizes the leftovers. It ferments sugars around it and pours out lactic acid, dropping the pH of its little patch. That sour acidic zone can make life harder for many harmful microbes. It does not stop at acid. Some strains also make bacteriocins, tiny antimicrobial peptides that disrupt rival bacteria. Acid plus peptides let a small transient population push back bigger threats. These weapons have names, and some are strain-specific. What is in the arsenal? Two main tools. First, lactic acid, the sour byproduct that keeps local pH low and the neighborhood harder for invaders. Second, bacteriocins, small peptides that can disrupt rival microbes. One cousin, Lactobacillus reuteri, can make reuterin, a broad antimicrobial compound. Together, acid and antimicrobials let a tiny passing population create an outsized defense zone. But if this microbe is so well armed, why is it barely a colon resident? And how do you keep seeing it? Lactobacillus is rare in the colon because it often does not truly move in. It arrives, works, and passes through. You top it up from the outside, especially through fermented foods where these bacteria can actively ferment. Whether a specific strain helps depends on the strain, the person, and the context, so this is not medical advice. The useful part is the handoff. The lactate it leaves behind can feed other residents that turn it into something even better. Defense is one job. Digesting your food is another. Next, meet the city's master chef, Bacteroides. You just met the bouncer that defends your gut with acid. But defense does not feed you. Digestion does. Meet Bacteroides, the city's master chef. It is a plump rod with rounded ends. and in many adult colons, it is one of the most abundant bacterial groups. Your body has only limited tools for breaking down complex fiber. Bacteroides carries a massive enzyme toolkit you do not have. Think of a chef that can cut vegetables your own knives cannot touch. So, what is on its cutting board? On the chef's cutting board is tough plant fiber. Bacteroides studs its surface with grabbing machines called polysaccharide utilization loci. They lock onto complex fiber, reel it in, and dismantle it sugar by sugar. Its scraps can feed neighbors, including butyrate makers. But there is a twist. In fiber-starved mice, some gut microbes switch toward grazing the mucus lining instead, thinning the barrier and letting trouble through. Mouse data, not simple human diet advice. So if Bacteroides can feed you and graze near you, what does it build? A chef is judged by what leaves the kitchen. Bacteroides makes two very different outputs. From shredded fiber, it releases short-chain fatty acids and smaller fragments that feed the community. One cousin, Bacteroides fragilis, makes something stranger. Polysaccharide A, a sugarcoat molecule that acts like an immune message. In mouse and lab studies, that molecule appears to coach immune cells toward tolerance instead of attack. So this chef feeds the city and writes notes to its defense system. What does all of that add up to? Bacteroides is one of the hardest working residents in your gut, but the same skills cut both ways. Inside the colon, it helps turn fiber you cannot digest into useful fuel and can help train immune tolerance. In mice, Bacteroides fragilis polysaccharide boosts calming regulatory T cells. Yet if Bacteroides escapes the gut through injury or surgery, it can become dangerous and is linked to serious abdominal infection. Helpful inside the walls, hazardous outside them. Same microbe, two faces. That idea breaks open next, because the most feared name in your gut is mostly misunderstood. You have heard this name in food poisoning stories. So why does your gut welcome it early? Meet E. coli, a misunderstood pioneer. Picture short, rounded rods drifting in the warm dark of a newborn gut. Many strains are harmless commensals, quiet residents rather than threats. E. coli arrives early, often before the famous founders take over. The scariest name in your gut may be one of the most misjudged. But why would the gut let it move in first? The answer is oxygen. A newborn gut still contains oxygen, and many important gut residents cannot survive oxygen well. E. coli helps solve that. It is a facultative anaerobe, meaning it can live with or without oxygen. Early on, it consumes oxygen and helps pull the environment toward the airless world that later anaerobes need. It is a pioneer preparing the ground for everyone who follows. No wonder scientists study it so intensely. E. coli became a window into life itself. So what does this oxygen pioneer leave behind for the city? Once oxygen drops, E. coli keeps earning its place. Some commensal strains build menaquinone, a form of vitamin K used in normal body functions such as clotting and bone biology. The careful point is this. Gut E. coli is not your whole vitamin K story, and food still matters. But the menaquinones it makes can also feed neighboring microbes with incomplete pathways, helping the anaerobic city hold together. This is the same organism scientists later borrowed to make human insulin in the lab, not in your gut. So where did the bad name come from? Why the fear? Because out of many E. coli strains, a few specific ones became dangerous by picking up toxin genes, often from viruses, that harmless residents do not carry. Those rare strains can reach people through contaminated food and make headlines. Same species, a few bad actors. The E. coli living in you is often a helpful kind, the oxygen pioneer and vitamin maker, kept in check by a balanced city. Keeping residents in check is a job in itself. Every city needs trainers to teach its soldiers. Train them too hard, and the soldiers can turn. Your gut wall is also a training ground. One recruit looks nothing like the others. It is segmented filamentous bacteria, or SFB. These are long beaded filaments that physically anchor one end to your gut lining and hold on. By anchoring there, they wake signals in the cells just beneath the wall. It is contact with a purpose, like the first lesson in a defense boot camp. But which immune soldiers does this trainer build? That is where the story gets interesting. Last part, the filament anchored to your gut wall. Now watch what that grip does. The attachment is not an attack. It passes signals into wall cells, and in mouse studies that nudge helps nearby immune cells become a specific fighter, the TA17 cell. Think of it as drilling a raw recruit into a trained guard, one that patrols the gut lining. A trained guard still needs a weapon. So what does this new soldier fire? Your new TA17 guard is trained and armed. Its key messenger is called IL-17. Think of it as a flare fired along the gut wall. That flare calls in backup and helps tighten the wall against invaders, especially fungi and certain bacteria trying to break through. At the right level, this is essential defense. Too little, and the wall is vulnerable. Too much, and the alarm can become part of the problem. At the right dose, immune trainers help build a gut that defends itself. But push the lesson too hard, and the Th17 response can overfire. In mice, overactive Th17 signaling is associated with immune attacks on the body's own tissue, including arthritis-like models. This is where we hedge hard. Human roles are still emerging, and these residents are trainers, not villains. Balance is everything. Some residents do more than train the body. Some whisper to your brain, that is next. You have met the trainers. Now meet the whisperers. Deep in your gut live quiet residents, including spore-forming bacteria and Turicibacter, a name worth knowing. Picture slender rods, some sealed inside tough little spores that help them survive hard times. They do not shout, and they do not control your mind. They sit near cells lining your gut and seem to murmur to them chemically. Scientists are studying whether that murmur can connect to mood biology, but before believing too much, we have to be careful. Here is what has been seen, mainly in mice. Tucked into your gut wall are rare sensor cells called enterochromaffin cells. Microbial signals can knock on those cells, and the cells answer by turning up their own internal factory. In one landmark study, gut bacteria helped that factory run at full speed. Germ-free mice made far less. This is a handoff. Microbe sends a signal, gut cell receives it, gut cell does the building. Notice what has not happened. Nothing has reached your brain. So what is the gut cell building? The gut cell builds serotonin, and here is the fact many people get wrong. Around 90% of the body's serotonin is made in the gut, but gut serotonin does not cross into your brain. It works locally, helping things like gut movement. It is not the mood serotonin in your head. What could link gut and brain is the supply chain, tryptophan, immune signals, nerve signals, and related metabolites. Some pathways may also touch calming molecules like GABA. The message that may travel is not gut serotonin itself, it is the system around it. So is your gut talking to mood biology? The honest answer is maybe, and the science is still emerging. In mice, changing some microbes can shift behavior and brain tryptophan supply. In people, we mostly see associations, not proof. Gut serotonin still never becomes brain serotonin. This is a whisper, not a switch. No microbe cures anxiety or depression, and this is not medical advice. A calm, well-fed gut may send steadier signals up the line. But when balance breaks, quiet residents fade and opportunists bloom. Not every resident of your gut is there to help. Some are waiting for the city to slip. Meet Bilophila wadsworthia, the bile eater. It is normally a tiny minority, lurking in the background. This oxygen-shy rod is drawn to bile, and bile can help it grow. In a balanced gut, it may stay rare and mostly quiet. But change the chemical environment, and you may hand this lurker a feast. What you feed the ecosystem can decide whether it stays quiet or blooms. Here is how a quiet lurker can become a problem. In some mouse studies, diets that increase certain bile acids also help Bilophila expand. One key molecule is taurocolic acid, which carries sulfur. Bilophila can use that sulfur in its anaerobic metabolism. The richer the bile supply, the more room it may have to grow, and a rare lurker can become a crowd. Most of this is mouse and association evidence, not proof of simple human diet cause. This is not diet advice, but what it releases next is the trouble. Every breath this microbe takes leaves something behind. As Bilophila uses sulfur chemistry, it produces hydrogen sulfide, the gas known for a rotten egg smell. In tiny amounts, the gut can handle sulfide biology. In excess, hydrogen sulfide can stress the mucus blanket and irritate wall cells. In studies, Bilophila blooms are associated with more inflammation, leakier barriers, and higher levels in some inflammatory bowel disease samples. Associated in mice and patient samples, not proven cause. One villain stresses the wall slowly. The next waits for defenses to fall, then erupts. Some villains do not creep. They wait, then erupt. Meet Clostridioides difficile, a gram-positive rod that armors itself in a tough spore. When antibiotics or illness wipe out enough of the crowded microbial city, C. diff can lose its competition. Spores wake, cells multiply, and the empty colon becomes vulnerable. Then it releases toxin A and toxin B, which attack gut wall cells and can cause severe colitis with intense diarrhea. This is a genuine medical emergency, not something to manage alone. If suspected, seek medical care. Next comes a trespasser linked to tumors. One microbe in this story does not belong here at all. Its usual home is your mouth, in the plaque between your teeth. Meet Fusobacterium nucleatum, the trespasser. Its name means spindle, and that is its shape, a long, thin, tapered cell, pointed at both ends. In a healthy mouth, it can be an ordinary resident. The trouble starts when it leaves. This needle-shaped microbe is built to stick, with surface grips that latch onto other cells. Researchers keep finding it deep in the colon, pressed near tumors. How does a mouth microbe get there? How does a mouth microbe end up near a tumor in the gut? Two routes are under study. It may travel down the digestive tract when defenses are weakened, or it may slip into the bloodstream through inflamed gums and ride to the colon. Either way, when it arrives, it does not simply float past. It anchors. Its needle tip presses against the gut lining, and surface grips pull it close enough to interact with host cells. It appears drawn to sugars displayed by wounded and cancerous tissue. So what does it do once locked on? Up close, the trespasser carries tools. One grip is a protein called FADA. Another, FAP2, helps it lock onto tumor-associated sugars. When FADA grabs a host cell, lab studies suggest it can poke growth pathways inside that cell. At the same time, Fusobacterium can stir inflammatory signals around the tissue. FAP2 may also dampen immune killer cells that would normally attack. This does not prove it causes cancer. It shows why researchers take it seriously. It can bind, inflame, and help hide in a risky niche. What does that add up to? Here is the careful part. In colorectal cancer, Fusobacterium often shows up more than in nearby healthy tissue, and high levels are associated with worse outcomes in some studies. A 2024 study found a specific substrain enriched in tumor niches. But association is not cause. We do not know whether it helps drive some tumors, simply thrives in them, or both. This is active research, not diagnosis and not medical advice. What it proves is the stakes. Who lives where in your body matters. The cast is not all risk. Some residents mark a healthier ecosystem. Of all the microbes in your gut, one appears strongly shaped by your DNA. Meet Christensenella, a small, oxygen-shy rod that gathers in tight communities deep in the colon. In twin studies, Christensenella was one of the most heritable gut microbes found, more similar in identical twins than fraternal twins. It does not work alone. It anchors a network of co-occurring microbes that often appear together. Genes may load the dice for how much you carry. But what does this inherited signature mean for the body? There is a gut microbe that often appears more in lean people and less in heavier ones. Christensenella ferments fiber and shares leftovers, including short-chain fatty acids the gut wall can use. Where it is abundant, it often sits inside a lean-leaning ecosystem. In people, higher levels are associated with lower body weight. In germ-free mouse experiments, stool containing Christensenella was linked to less fat gain than stool without it. Associated in people, mouse supported in experiments, not a treatment and not guaranteed. You cannot bottle this signature yet. Another microbe tells a different story. What you eat. Some microbes can almost read your diet. If Prevotella is thriving, it often points toward fiber-rich, plant-heavy eating. Prevotella feeds on complex carbohydrates and is common in traditional plant-based dietary patterns. But here is the honest twist. It is not purely a hero. Some Prevotella copri strains have been associated with higher rheumatoid arthritis risk in certain people, and the link remains debated. Same genus, different strains, different context. So this is not good or bad. It is ecology. Which raises the real question: how do you tend a garden this complicated? You have now met the cast inside your gut. Founders, peacekeepers, gatekeepers, protectors, chefs, pioneers, trainers, whisperers, opportunists, trespassers, and ecosystem signatures. Here is the truth: none of them can change alone. You cannot buy this whole cast in a bottle. No pill rebuilds an ecosystem by itself. But you can feed it. More plants, more kinds of plants, and fermented foods can feed more kinds of microbes. Genes set the stage, but every meal helps decide who eats. Tend the garden, and this cast tends you back. Now you know who lives there.